This will give me the button. Sorry, just move everyone out of the way. Right. So hopefully I have managed to share my screen without displaying my emails or displaying it in presenter mode. So welcome everyone. So as Julia said, I am the new director of the Christabel Pankhurst Institute, so very much finding my feet here. Still, as I am speaking, I am sat in a room on my own. So do just ask questions. If anything's not clear, you can put your hand up, but I might not see that. So yeah, if you've got an urgent burning question and you're happy to be on the recording, then please do step in. So I thought I would start by saying, what is the Christabel Pankhurst Institute? So because this is sort of kicking off this series of seminars, it's probably just worth revisiting for all of us what the Christabel Pankhurst Institute is. And this is and that's a very good question, given I've only been the director for 3 weeks. But we're here to support sort of research and innovation that sort of needs-led looking at health technology, and we can specifically look at supporting that research through the translation pipeline until actually getting to practice. And hopefully we can act as a external beacon for collaboration. So that primarily could be with the foundation trusts nearby, but it could be with other key stakeholders around the country or internationally. So or industrial partners as well. So we're here to sort of support your research moving through from the sort of original needs-led concept through to sort of delivery into practice. And we are set up to be sort of challenge-led. So hopefully a lot of the work that I'm about to talk about could fit into either any of these 4 buckets. But we've got health equity, any sort of questions around health equity, and make sure we've got sort of public health challenges there, an inferential challenge around prediction and diagnosis, a sort of physical tech challenge around interventions, and thinking around the digital ecosystem, thinking around how we can deploy tools regionally to sort of optimize patient pathways or patient outcomes. So yeah, we have these 4 challenges. I feel that these are sort of natural and intuitive challenges, but we are going through a process with the other deputy directors, given we're all relatively new in post, just to explore some of this. So some of this might change over the next few weeks or months, but yeah, this is kind of how we're currently sort of set up. And crucially, we have these sort of programs, but more practically, perhaps, rather than these sort of relatively dry statements about how we're set up, we have physical support. So I guess the low-hanging fruit for that is that we have event spaces. So we've got sort of physical meeting rooms, but there's also the Emmeline suite next door to where I'm sat at the moment for having collaboration events. We can foster interdisciplinary research, and we have the translation toolkit that you can sort of use to sort of look through your research and innovation demands through the pathway. And so I encourage anyone to sort of have a look at the translation toolkit that Claudia and the team have been working on over the last few years. So that's how the Pankhurst is sort of set up and can potentially support your work. What we have, what I'm going to talk about today is some work that I did pre-pandemic in a previous job when I was working at Public Health England, and I led the emergency response analytic team. And so this slide is one that I would often give in talks at that team, and this is how sort of showing how we were set up there. We would have these pillars of reverse epidemiology and contingency planning. That's essentially a fancy way of saying statistical inference and applied mathematics. And they would feed into decision support, so sort of operational research and sort of translation of those ideas to policy. And then that would be supported by high performance computing, quality assurance programs, and collaboration networks. And then we'd have this sort of underpinning work stream to improve our understanding of what the mechanisms were that fed into the modelling. So that could be eco-epidemiology if you're looking at vector-borne diseases, the dose response if you're looking at bacterial challenges, or behavioural response because people have an annoying way of reacting in ways that you are not predicting they'll react in. So this was very much how we were set up there then. And it actually resonates quite nicely with those challenges on a couple of slides ago, in terms of sort of computational statistics and computational sort of applied maths, and then translating that through to practice. And some of these images around this slide we'll come back to. If I can get there we go. And this is very much, as we're thinking about translation, this is very much an iterative journey. The schematic here is, I put this in because this was, I've just recently taught the first year stats lecture on defining what statistics is. And actually this sort of schematic is quite helpful for framing analytic modelling, perhaps more generally around sort of collecting data, organising data, analysing data, interpreting data and presenting data. But it's really a helix. And so as we move up the technology readiness levels, that data gets more sophisticated, the models get more sophisticated, and we sort of keep going through this iterative journey. So one of the sort of the title was fairly euphemistic about emergency response. The sharp end of what we were doing was bioterrorism preparedness. And so it's easy to sort of think, oh, well, there's not much threat from bioterrorism because there haven't been that many bioterrorist events. But these two on the screen are two sort of foundational near misses from the Sverdlovsk outbreak in 1979, and then the cult dispersion in the 90s. And what sort of happened with these two, it wasn't a terrorist event as such, but in the Sverdlovsk outbreak, I'm sort of slightly going to paraphrase this, but essentially what happened was they did some cleaning of their manufacturing plant for weapons grade anthrax, and they forgot to put the HEPA filter back on the on one of the emissions bits, and ended up disseminating the anthrax over the local city. And I forget exactly how many cases there are. I think the picture there gives some of the case numbers, but they pretty much fall downwind for a particular day. And so Messelson and colleagues, when they analysed this post hoc, were able to show the wind was blowing on one day, going over the entire cloud of people, which suggests that they realised their mistake with the HEPA filter quite quickly and put it back. The Japanese cult is quite interesting because the paper there with molecular investigation into the release That is the entire abstract for that paper. And it understates things there's quite a lot unsaid in that abstract. So the Stern 3,4F2 is the anthrax vaccination strain that they use in animals. So they did release anthrax over the city, but they released the vaccine strain. So they essentially vaccinated the population of the city. Now, and I mean, you could go into the whys and wherefores of that, but the other thing that's quite interesting here is that no one noticed. So that 1993 date is quite important because the other key event that this cult is associated with is the sarin attack, which happened in 1995. And it's my understanding that it was only in 1995, after the sarin attack, that the police went and investigated the headquarters of the cult, raided it, and essentially found this brew of anthrax on the rooftop, venting out. So, I mean, you could class that as a near miss or what have you, but these things sometimes go undetected, or it could, yeah, the causative action, sorry, is quite varied potentially. And then, so those two are quite big releases: a terrorist action and a sort of cock up in a factory. But then there was this bongo drumming, African bongo drums, sort of flurry of outbreaks in the 2000s, where essentially anthrax is present on the goat hides that the bongos are made out of. They're imported in. They're not necessarily cleaned very well. And so they make the bongos, and then these people go off and do bongo classes in village halls around the country. And so one case, the bongo drummer was infected from this, from their drum. But the 74 people that attended the classes, and then the 400 users of the village hall subsequently, did not get infected. So that tells us possibly something about dose response, or exposure risk, although it can be quite difficult to sort of really be too clear cut on that. But and then you have there's sort of an interesting corollary to that, where there was the village hall users, one of the Scottish village halls, that the villagers really didn't want their village hall demolished and rebuilt, and in terms of clear up. So sometimes the clear up activity could also be quite difficult to manage. And then moving away from anthrax, we have another potential bioterrorist agent, tularemia. There were some outbreaks of tularemia in Martha's Vineyard, and they did this case control study where they showed that lawn mowing and things were associated with illness. And then when we originally read this paper in the early 2000s, we were like, why are they focusing on lawn mowing? And then we read their citations, and they cite this lawnmower tularemia case outbreak, a sort of single, well, it was only a couple of pages long. I've just highlighted one paragraph where you can sort of imagine these two kids in hospital were being interviewed about how they got tularemia. At this point, the boys admitted running over a rabbit with a hand-push lawnmower. You can, even though, I mean, they didn't necessarily see it was there, but then they rerouted it afterwards. Sorry, is that someone else? Is that a question, or is that just an unmuted mic. No, okay, I'll assume that's just an unmuted mic. Yeah, never underestimate how people can infect themselves with some of these agents. But if we'd seen a couple of cases of tularemia in the wrong place at the wrong time, we might have associated that with a terrorist attack when actually it was people doing silly things in their gardens. So how can we think about this problem of the more holistically than sort of, and turn these ad hoc events to something a bit more systematic? Well, we use the qualitative, sorry, not qualitative, got it wrong straight away, quantitative microbial risk assessment approach. And so that means breaking down the scenario into these subdivisions. So we've got the source term, worrying about how the source works, then dispersion, where it goes, the dose response, who it infects, the incubation period, how long between cause and effect, the epidemic curve, which is the effect, and then thinking about how we control it. Now, the source term tends to be a bit disconnected from it's quite hard sometimes to actually see the source. So it can be a bit disconnected from the wider thing, or it's a bit approximate. It tends to be quite, if you're doing a scenario-based thing, it tends to be quite sensitive. And you have to decide whether you're going to sort of worry about it being covert or overt. So did people notice a cloud of stuff, or was it the right time of day or whatever that people didn't notice until they turn up in hospital? When I gave a similar slide deck a few weeks ago to a workshop in Cambridge, I said this tends to be a bit secret squirrely. And then everyone in the room looked at me blankly because they're all younger than me. And I'm not this old, just crucially, but they used to have reruns of Secret Squirrel cartoons in the 80s. So yeah, there was this spy squirrel. So when I say Secret Squirrel, I'm sort of, it gets a little bit you start getting into spy realms if you're not careful. So we tend not to worry. We tend to either just learn from the data what could be a potential source term, or we have very simple source terms. But this does lead to questions about what happens if it was 5 releases very close together, as opposed to 5 attacks at the same time in very different cities. And so the sort of dissemination of, but you'll be able to tell that from the spread of cases if you're careful. Dispersion modeling, you can do computational fluid dynamics, so CFD, if you really want to, if you have the computer power. Often it's simpler to approximate the spread a little bit more with just a turbulent advection diffusion model. So yeah, apologies to people who are listening that aren't used to seeing equations, but this is just putting the equation for this Briggs model on the screen to show. And I think the thing to take home from this is this turbulent advection diffusion model, this Briggs formulation, was originally developed for large-scale nuclear releases and validated against trials that were conducted in the desert in Nevada and places in the 50s. And the sigmas, the sigma Z and the sigma, the sort of lateral spread, or the diffusion on this spread off the downward direction, they are functions that were just empirically defined based on flow from a desert, around a desert. So we are sort of mapping that, we're just sort of translating that to urban settings or different terrains. And so we do need to have a bit of pinch of salt with some of these, the fidelity of some of these models. And that would be true if you were using a complicated CFD model as much as these simpler models. And equally, people very annoyingly do just move around within that cloud. They don't just stay still. So the exposure can be a little bit unclear as well. So we have to be careful as modelers that we're not being over reliant on the model. But this is where interdisciplinary collaboration can be really important, because we need to think about the environmental modeling, the METs, how good the MET is, and sort of work with colleagues to sort of tweak some of these models and just sensitivity check some of the assumptions we're making. Similarly, with dose response, we looked at the anthrax dose response. A lot of the anthrax modeling done in the early 2000s was based on this competing risks framework, that you have some germination rate of the spore when you inhale it, and then your body clears it by some process. And statistically, you can work that through, and it's just then boils down to a single parameter. Now, but you can take that, you can make that a little bit more complicated, that you have variation in the host. So everyone is different. So their clearance rates or the germination rates will be different. So you can factor that into the models. You can allow for clustering of particles in the cloud that people are inhaling. And you can allow for people doing different activities. So there's variation in their inhalation rates. So some people will be running around, some people sitting down, some people asleep. And you can come up with various functional forms for this dose response function. However, it still just looks like a sigmoidal curve. And so actually the quality of data we often get isn't sufficient to nail down a particular model. And this is an example of that. So this is looking at the probability of death in guinea pigs that were given a certain dose of anthrax. So the black triangles are the guinea pig data, and all of those models, the smooth curves, the solid, the sort of coloured curves, are model inferred sort of estimates, smoothing that data out. Now, all of them are pretty much similar fits to the data statistically. You can't use statistical tools to learn which model is more appropriate. And that, I mean, so that kind of leaves you feeling, oh, we could use any of them. And that is true to a certain extent, but the problem is extrapolating out of the sample. So if we take the left-hand curve and blow up the axis and look at low doses on the right-hand panel, we've got no data, but this is just drawing these lines back to lower and lower doses. And you can see the viable fits, the orange dotted line, is orders of magnitude greater than the exponential or the other fits here. And the blue line, the exponential, is the competing risk model fit. And so the mechanistic model that you're fitting can really impact on your inference. And the reason this low dose is important is, as if you take a city and you take that dispersion cloud over the entire city, you're going to be hitting thousands and thousands, perhaps millions of people, all at the sort of low dose end of the spectrum. So whilst it may seem that you've got low risk, you're multiplying that risk by a huge population. So actually you'll be massively overestimating your cases if you're not careful. And so we do need to have some sort of mechanistic understanding of the rather than sort of a heuristic sort of understanding of some of these mechanisms. So we do just need to work multidisciplinarily to learn this. Um, and then another thing that's kind of unhelpful is in the literature. So this is some work that Jingxi did with me on COVID, looking at the dose response to COVID. But the point, so I'm jumping around a little bit on the causative agent, but the important thing to take home here is that in the literature, people are calling the functional form that I'm put on the slide there the beta Poisson model. But the beta Poisson model, if I go back a couple of slides, is really this 1F1 function here, the hypergeometric function, not this function with the news and stuff in it. And that's because this is an approximation to the beta Poisson model. In a particular parameter coupling, you've essentially lost a parameter. Someone has done some analysis at some point in the literature and derived this function, but then just called it the beta Poisson model, whereas really it's the approximate beta Poisson model. And you can't guarantee that the approximation is always true. So you have to be careful what the literature, sometimes how people in the literature are using the maths to draw their conclusions. And then there's the point, this is again sort of leveraging some of Jingxi's work on COVID, but you have to be careful. So you can come up with quite arbitrarily complex models for the in-host dynamics post-infection. So you have cells, they get invaded by the virus or the bacteria. We've got similar models for bacteria. Then there's some sort of T cell response or some sort of adaptive response that grows over time. But essentially, you've got two mechanisms that you can't really differentiate. There's the depletion of your susceptible cells and the growth in the innate response. They're doing the same thing. So from a modeling point of view, you can't really learn the mechanism. So you have to work with people who do understand the mechanism to work this model through. And you can and if you do that, or if you get if you simplify the model, then sometimes you can get relatively simple forms out for those models than having to solve many differential equations. You can't always do that, and sometimes that is not a helpful way of viewing it, but this is where kind of a little bit of analysis of the system can help. I will, in the interest of time, I'm just going to sort of skip over the incubation period, but what is important here is we as mathematicians like to use the gamma distribution to model the incubation periods because there's nice properties that allow us to do things with ordinary differential equations, but in the literature, people often fit any skewed distribution, so the variable, the log normal, or the gamma, but then often they don't really count the fact that people report their incubation period. So when they were exposed to when they develop symptoms, often that's reported to the nearest integer day because they just remember difference in dates. So there's sort of an inherent censoring. So again, sometimes you have to be careful how you interpret some of these models. And we had a PhD student who's recently finished, Niall Jameson. He and gone to Texas, had a look at some of these models with alternative models for the incubation period that seem to be slightly better, but you have to be careful how you're defining better. So I've spoken quite a lot there about models and some of the sort of structural assumptions. If we just think, and I've spoken about sort of applications to non-transmissible diseases, sort of anthrax and tularemia. Plague is another interesting example of potentially bioterrorist. Another potentially bioterrorist agent, accessible agent. It exists. You don't want to be in prison in Madagascar, and you get the occasional case in Nevada, I think, as well, in the US. But the interesting thing about plague is that yes, it's sort of famously this sort of rat, flea, human epizoonotic cycle, but there is a form of plague where people start coughing. It's a complication of plague where people develop a pulmonary infection, start coughing, and then can spread person to person. And we've seen outbreaks of that where I think the last few cases in England were in 1918 in Anglia, and it's just like a few houses in a village, but the you can see the social cultural impact of the disease because it was in one household, the children got infected, they died, the mother got infected looking after the children, they died. When the mother died, another woman from down the road came in to help look after the children, and she got infected but took the infection back to her house, and so on. So we were able, but there's not many good outbreaks of good data sets for plague. We stumbled across this data set from 1800 in a report that I stumbled across about Napoleon's expedition to Egypt in the late 1700s. So he took an army to Egypt. He tried to work up to Jaffa. Well, he got as far as Jaffa, trying to invade the Ottoman Empire, and then plague stopped him. There was a plague outbreak. He went back to Cairo. By the time he got back to Cairo, Nelson had sunk most of his ships, so he ran away back to France, and then became proconsul or first consul in France. But he left behind his medical military doctor, who, with little else to do militarily in Cairo, started counting the deaths in the local population, and so we have this time series of mortality in Cairo, which we wouldn't otherwise have had. And we can fit models to that, allowing for the rats and the fleas and the humans, and we can fit those models to that data. Now, an interesting point on this is that I presented this. This was done for a US collaboration, and the US policy teams said, we can't show this to our policymakers. We can't, sort of decision makers. And he said, why can't you? And he said, well, some of your data's outside of the confidence interval. And so we sort of realized we were talking at cross purposes about prediction intervals and confidence intervals. So you do have to be careful how you communicate sort of some of these ideas as well, and that's something hopefully we could help people with. And then there are the sort of ad hoc outbreaks, a plague in Johannesburg that a historian found and came to us trying to analyze, and so we're able to look at the transmissibility of that plague outbreak using an established tool. But the point here is we're sort of being a bit ad hoc. We're just analyzing the data that we can find. We probably need to be more systematic in how we find some of this data that's lurking away in the epi archives, and try and sort of more systematically fit some of this. And some of that's being done by the modeling community through Epiverse and such like. But yeah, being more systematic would be good. So we've sort of got through to the epidemic curve. Some of these tools for the analysing epidemic curve are very similar to the ones we would have used in the pandemic, or sort of slight adaptations on the ones we would have used in the pandemic. Here, for a non-transmissible disease, essentially all we're doing is fitting the incubation period, and so we can do that. What we're looking at here is a spaghetti plot rather than on the right-hand side, rather than a confidence interval plot, they're essentially showing the same information. It just shows a little bit more of the variation. So sometimes it can be a bit clearer to policy teams why there is difference in the outputs than burying everything in a confidence interval. But sometimes that doesn't really work as well. And then space. So when we were building up for the Olympics in London in 2012, we had to make sure that we were geared up in case there was a terrorist attack at the Olympics. And so these tools were tested. We had various exercises at very senior levels to make sure people could understand it. And so what you're seeing here is some outputs from some of the papers we had in the sort of mid 2000s. So they're very much 2009 graphics. But we could sort of simulate an outbreak. This one going out over the Thames estuary, and then look at countermeasures. But that was decided that we needed something a little bit more sophisticated for the Olympics, and so we had a different scenario, and we developed what we call the countermeasure delivery optimization tool, which essentially, given a hazard footprint, so sort of the dispersion of cases, it would calculate which boroughs or which regions of London or spatial areas of the UK you should target for countermeasure deployment in the next 24 hours or 24-hour blocks after that to optimize the response. So to minimize your potential number of deaths. And so the red zone that you can barely see on this plot on the right-hand side, that was the zone that was inferred to be the area that should be given its countermeasures first, then the orange zone, and then the yellow zone. Clearly, we would expect the information to change, and so the next day, having done the red zone, there might be a slightly different wind direction or something, and these areas change. But that's the sort of tool that we had developed, and hopefully we will be able to take this work forward in the next few years with a new PhD studentship. But we also worked with DSTL on the Crystalcast project, which eventually pivoted to working on the pandemic. But before that, we were looking at optimal control for bioterrorism responses, and we were working with David Spiegelhalter at Cambridge, trying to work out how you present some of this information to policy teams or to the public, because once it goes to the policy teams, it has to go to the public for them to sort of make sense of things. And so we were trying different visualizations for that, using these sort of charts. So any model is just an abstraction of the real world, and we can be arbitrarily complex, but we're often restricted by the availability of data, and that data could be to generate hypotheses or to identify mechanisms. So working interdisciplinarily is absolutely critical. What often we don't know is the sort of trigger mechanisms. The causative agent may not be known. It probably is quite clear if it's a pulmonary, if people are in hospital with pneumonia. So once the symptoms are known, it's probably a bit easier. Chemical releases would probably be a bit faster, probably a bit more overt. As just before the pandemic, I was involved in an exercise at the EU in Brussels, and they threw into this scenario a hybrid attack. So you had a CBRN event and then a hack on the hospital infrastructure, IT infrastructure. So the IT went down in the hospitals. So you can sort of create these sort of arbitrarily complex hybrid risks if you're not careful. And we have, excuse me, a the how clean is clean worry as well. So if there was anthrax sprayed over a large area, you'd have a re-aerosolization risk. Modern technology for diagnostics of anthrax in the environment is getting better, so you'll be able to find it more easily. And there is a very real risk that companion animals would be sensitive to certain infections. And so, yeah, I do you have to worry about not only the human but the veterinary side of things. And I think these pictures are from the Novichok outbreak. I put them on here just because it's easy to sort of dehumanize these outbreaks, as we sort of said on some of those early slides about the Japanese cult or the Russian factory, and not have these. But this happened in Salisbury, and whilst I was working in Manchester, I had gone to one of these exercises in Brussels. And we were still living in Salisbury, so I had been in this market square where the police car is on the Sunday with the kids to go to the library, which is pretty much just the other side of this police tape out of shot. And then I flew off and woke up in Brussels, turned the telly on and find this all over the press. So suddenly you sort of, do get a sense of the human angle and the sort of interconnectedness, perhaps. So I just wanted to spend a few minutes. I've spoken very much here on the bioterrorism events and work that I'd done previously, but I just wanted to have a few slides on the pandemic response. So we obviously had the pandemic through 2020 to 2022. A number of us were heavily involved in that response in different ways. I got pulled into looking at care homes and to a lesser extent prisons, and how you control infectious disease outbreaks in those settings. And so hopefully we have some grants under review, and hopefully we'll get them good news soon from UKRI that will give us some sort of platform for taking some of that work forward, looking at the whys and wherefores of outbreaks. And I think the thing we've learned there is you need to think about the holistic picture of the response. And so it's not necessarily just about the health effect of the physical intervention, but how that could impact on the population at risk. And so if you isolate everyone, is there a sort of psychological impact on people that is worse than the risk itself from the disease? And so we need to find ways of collecting that data and actually having something objective to look at. Otherwise, it'll always just be a subjective argument. And I probably don't need to remind everyone how sort of I won't sort of go into lengths here, but the SAGE process, this would be similar if there was a bioterrorist event, probably less groups, but it would still set up as SAGE being the group for scientific advice in emergencies. They would be supported by subsets, and then SAGE puts the advice forward to COBRA, who make the policy decisions. So all of this scientific advice is just advice. We're not setting policy here. And this can get quite complex quite quickly. From a pandemic perspective, you're trying to look at how you intervene. It's a very complex parameter space. We'd like to deal with as few parameters as possible, but the interventions, what your intervention is often only really controlling one aspect of the disease pipeline. And so you probably need multiple layers on your intervention. There's the sort of conceptual frameworks like the Swiss cheese model, that you need these different layers so that things can't get through the entire pipeline, rather than some magic bullet. Although vaccine is almost that magic bullet that it cuts through a lot of these, but you still need to layer vaccination with other things. So I will, I think, stop there and take any questions. And apologies if you have had your hand raised for ages. So I'll come back to the room. Any questions?